Hello, everybody. Thanks, Evan and the organizers for the invitation. This is such a great meeting and such a great initiative. I'm happy to be here. I will talk about initiatives in our lab of trying to model mood disorders using stem cells from patients. The motivation for a lot of us at the disease modeling field using stem cells is a lack of good drug candidates to treat neuropsychiatric disorders. The truth is that over 90% of drug candidates fail at clinical trials due to issues in safety and efficacy. And a problem for new therapies for mental conditions is not only the lack of compounds, but also the lack of basic understanding of disease biology. And we think that harnessing knowledge that is already out there, such as good genetics, as Kristen has mentioned before, good genetic analysis, brain imaging, good clinical assessment data, postmortem tissue analysis, and rodent models, together with stem cell disease-based models, it's a way to help gain more biological information. So today's talk, I'm going to talk about mood disorders, taking patient samples from bipolar patients and reprogram those, differentiate them into progenitors that can be either neuroprogenitors or glial progenitors, and then studying either the effects on the neurons or the effects on the glial cells. Our approach, as have been mentioned before, there is a great heterogeneity between different patients and different populations, especially when you're trying to do disease modeling in a dish. So our approach was to take bipolar patients that were a cohort that were either responsive to a typical drug treatment for bipolar, which is lithium, or patients that were non-responders to the lithium treatment. So we reprogrammed them into IPS and then into neurons or astrocytes, treated them with lithium, and tested the neuronal function in response to the drug treatment. And our hypothesis was if patient neurons will respond to the treatment if the patient is a responder. So we set up about 3 years ago, to test this one hypothesis. And we used the cohort of patients from John Kelso at the psychiatry department in UCSD. And John had 4 neurotypical controls. We got fibroblasts from these patients, 3 lithium responders and 3 lithium non-responders. And this is the work led by Jun Yao and Jerome Mertens back in 2015. And what we adapted a protocol of making PROX1 positive neurons, which are dentate gyrus fate committed, and we differentiated our stem cells using a protocol developed by Diana Yu in our lab in 2012. And Roberto Giappelli was a postdoc in our lab at the time, developed a marker, a reporter, that was PROX1 promoter expressing GFP. So we were able to track those cells in vitro. Jun Yao then patched those neurons from bipolar patients that were either responders or non-responding to lithium treatment. And what he noticed at first was that the hippocampal dentate gyrogranular cells from bipolar patients show hyperexcitability. And he measured that by both spontaneous as well as evoked action potentials. More interestingly, he also noticed that lithium treatment changed the hyperexcitability only on patients that were responding to lithium. Then another postdoc came to our lab that wanted to see if that was true in a completely different cohort of patients. So Shirley Stan came to our lab at the same time that we were in contact with Anne Bang from Sanford Burnham Institute, SPP. And she had this well-characterized cohort of IPS derived from patients selected in Canada by Martin Alda. They were also responders and non-responders to lithium, but they were from a completely different cohort. So Shani set up into this adventure of looking at neuronal activity on those cells. And another additional change was that those patients gave us the somatic tissue to begin with. The process of reprogramming was not fibroblast, was lymphoblast. So that was a completely different cohort and a different cell source. So Shani was excited when she got here, so she was happy to do a lot of work, and she patched cells from those patients. And she also noticed that hyperexcitability was present in patients with bipolar, both on responders as well as non-responders. And what was interesting too, the different cohort but same response happened when you treat cells with lithium. Only patients that are lithium responders decrease the hyperexcitability, but non-responders do not respond to lithium. So lithium treatment reduces hyperexcitability only in neurons from the lithium-responsive patients. We thought that this was a good way to test that what's happening in vivo can actually be reproduced in tissue culture. For disease modeling purposes, that's always reassuring when we see commonalities. Then she became interested in looking if other cells, other neurons within the hippocampal area were also niche, were also affected by hyperexcitability. At that time, Anita Sarkar in our lab had developed a protocol to make enriched populations of CA3 neurons, and also we were able to identify them with a specific promoter. In that case was Greek4, a promoter expressed in GFP positive cells. So then we differentiated our cell lines, bipolar responders and non-responders, and controls using the CA3 protocol. And when Shani patched those cells, here on the right, she also noticed an increase in hyperexcitability on CA3 neurons, which was somewhat expected, but we wanted to see if that was the case. Those cells were also responding to lithium at the responders population, but the non-responders did not respond to lithium treatment. So that was interesting, and that prompted us to ask another question. Are neurons from the hippocampus the only ones that are hyperexcitable in bipolar patients, or neurons from other fates can also be hyperexcitable? So we tried to go as far as we could from the brain, and we went, and we used the protocol that we had perfected in a lab many years ago to make motor neurons. So we made motor neurons with our bipolar patients, using bipolar patients and controls. And here we also had developed modified the reporter that was expressing GFP under a motor neuron promoter called HB9. So Shani was then able to patch those cells, and that we didn't see any difference in the percentage of motor neurons for each patient. But interestingly, there was no significant changes in cell excitability, and that showed to us that hyperexcitable phenotype was specific for cells, at least in the hippocampus, data gyrus, as well as CA3, but not in motor neurons. So hyperexcitability is present in hippocampal neurons, but not in motor neurons. So to summarize this part, fibroblasts as well as EBV immortalized, so those are the lymphocytes, two different cohorts, two different patients, show hyperexcitability and can be differentiated into DG and CA3 motor neurons. DG and CA3 derived from bipolar patients are hyperexcitable, but motor neurons are not. And lithium treatment reduces hyperexcitability in neurons derived from patients who respond to lithium therapy. So we got interested then in look at other cell types that were present in the niche and see if there were any other changes in cell phenotype. And we looked at glial cells. Mood disorders have been shown to, in patients, to show increased levels of inflammation. So specifically in BD, increasing levels of pro-inflammatory cytokines have been shown in patients, and also increased excitotoxicity. And maybe that was an interesting way to start teasing those things using different cell models. So we started by developing a protocol in our lab that was making, with the idea to make inflammation-responsive astrocytes from human pluripotent stem cells. So at first, this protocol was derived by Renata Santos and Krishna Vadodara in a lab. And the idea was to create a progenitor population that was a glial progenitor population that we can expand and then freeze and differentiate into astrocytes. So we did that, and we wanted to show that those cells expressed astrocyte markers as well as glial progenitor markers. And we also show that the cells were able to perform glutamate transport, which is one of the hallmarks of astrocyte activity. And those cells can uptake glutamate. And we've also shown that they show slow calcium transients that can be blocked by gap junction blockers. And those were just showing what the control iPS-derived astrocytes were able to do. Our next question, this has been published last year at Stem Cell Reports. So we showed that iPS-derived astrocytes are functional. Our next question was, can they support neuronal activity? So what we did is we did a culture system where we differentiated NPCs, neuroprogenitors, into neurons, and glial progenitors into astrocytes, and we co-cultured them together in a platform called multi-electrode array, as has been talked about already by Kristen. So the idea is that we have electrodes that's from Axion Biosystems. We have electrodes embedded into the plate, and the electrodes are able to detect extracellular neuronal activity. So the readout will be spikes. So it's a spontaneous activity from the neurons. And when you look at the different units, you can have an idea of neuronal activity within your plate. And those are multi-well plates. We use usually 96-well plates, so we can look at many controls and many bipolar cell lines at the same time. Oops, other way. So what we noticed just using control astrocytes is that if we increase the amount of astrocytes giving a neuronal culture, the neuronal activity increases, which is expected but good that we can reproduce in our lines. So the more, if we increase the amount of astrocytes, we have activity. So the iPS-derived astrocytes improve neuronal function and increases the number of spikes and bursts. Then we got to our real question, which is, are we able to generate a population of astrocytes that is sensitive to inflammation, to inflammatory cytokines? And what we did is we adapted a FACS-based assay to stimulate, so now we stimulate the cells with either I1 beta or TNF-alpha, those are iPS-derived astrocytes. And we treat them with protein transport inhibitors, so then the cytokines are kept inside the astrocytes. And then we wait until 5 hours, and we count the percentage of cytokine-producing cells by flow cytometry. And what we see here on the right is that when you treat the cells with either I1 beta or TNF-alpha, the percentage of cells expressing either IL-8 or IL-6 increases. That's a shift towards the right. So we tested a lot of controls, astrocytes, to show that the activation was comparable to the first bar here, which is a primary astrocyte. So those are fetal extracted astrocytes, primary cultures. So the levels of activation is comparable to primary astrocytes. And then we wanted to apply that protocol to our patient cells derived from bipolar and controls. And we used our initial cohort from John Kelso comprised of 4 controls and 6 bipolar patients. And we performed the same cytokine array with those cells, and we analyzed the percentage of cytokine-producing cells after stimulation with either I1 beta or TNF-alpha by flow cytometry. And here we detect an increase of IL-6 expressing cells after treatment with either I1 beta or TNF-alpha. Because we had a bimodal curve, I wanted to plot lithium responders versus lithium non-responders within the plot to see if there was a difference. And it doesn't seem that there is a specific difference for responders or non-responders, at least for astrocyte activation. Because those were FACS assays and we're keeping the cytokines inside, we also wanted to know if cytokine release was different. What we did for that is we performed the cytokine array using media from stimulated astrocytes and blotted into membrane that contained pre-blotted cytokines. And when we did that, we noticed that once you treat the cells with vehicle, there is a certain number of activation. Those are control dots. But when you treat them with I1 beta, overall you see an increase of cytokine production. Those are 32 cytokines plotted here. But I just want to call the attention specific for IL-6, which seem to also be increased release into bipolar patients. We are further quantifying and analyzing this data and finding out if there are other cytokines that we could check. We're currently also looking at RNA-seq to see if there are specific differences in expression profiles. So iPS-derived astrocytes in bipolar patients show increase on the percentage of IL-6 producing cells after stimulation and increase in IL-6 release in the media. We then wanted to know if lithium had any impact on activation and if there was difference between lithium responders and non-responders patients at the astrocyte level. So we basically did the same experiment, except that we treated the cells with lithium or not. And we compared the percentage of cytokine-producing cells. And when we look at just control lines, we don't see a difference after lithium treatment. Percentage of cytokines is still as high as they were before. And when we look at bipolar patients, we still see a percentage of cytokine higher than controls, and lithium didn't seem to, change. I guess I should have looked at you earlier. I'm almost done. We plotted responders to see if there was a difference on the bimodal distribution, and we also don't see a difference. So lithium responsiveness does not seem to be playing a role on inflammation levels. Lithium does not affect the percentage of IL-6 produced. Lastly, we wanted to look at co-cultures, and here we just kept the neuronal population stable, same neurons, and we just changed the astrocytes that we were co-culturing those neurons with. And we used either control or bipolar astrocytes, and we pre-treated them with either vehicle or I1 beta. And again, we looked at activity using MEA platforms. We compared control versus bipolar, control versus control with I1 beta, bipolar versus bipolar with I1 beta. And here is just to give you an idea of the comparisons that we make. When we look at vehicle treatment and we look at the controls only, as expected, when you add astrocytes to neurons, again, population of neurons is always the same, is fixed. When you add astrocytes, activity increases as expected. When you pre-treat the astrocytes with I1 beta, and here is just pre-treatment, the neurons don't see I1 beta. The neurons just see astrocytes that have been cultured with them. Decrease in activity after I1 beta pre-treatment. And at the BD level, when you co-culture the same population of neurons with bipolar astrocytes, we see a decrease in activity. And then when we pre-treat those bipolar astrocytes with I1 beta, we see an even bigger, stronger effect and decrease in activity. And what I find really interesting here is that when you compare the control-treated co-cultures with control astrocytes treated with ion beta, that's actually comparable to bipolar. So it's almost like bipolar has this inner inflammation levels. Another control we did here, and that's in collaboration and with Ginny Jones and Bia Freitas at Cellular Dynamics, now Fujifilm, because our neuronal cells are differentiated from a neuroprogenitor population that can carry over some astrocytes during the differentiation. We wanted to use a pure population of neurons. And at CDI, they have pure populations of glutamatergic neurons. So we got a vial of pure population of glutamatergic neurons and plated that on the MEA and co-cultured them with astrocytes. And we observed a very similar effect that we had before. So we are confident that what we are seeing in activity is not due to residual population of astrocytes that we're bringing. We are actually seeing an actual effect. So two different populations of neurons showing the same effect. So to conclude, neuron activity decreases in co-cultures with astrocytes that were pre-treated with ion beta. Neuron activity decreases in co-cultures with bipolar astrocytes compared to controls. And decrease in neuron activity is independent of pre-existing astrocytes in culture that might carry over. So I already discussed the summary over my talk. I just wanted to highlight that our next steps would be to investigate the IO6 on neuronal activity in the context of bipolar. And in conclusion, we think that inflammation-sensitive astrocytes can be used to study neuropsychiatric and neurodegenerative disorders, conditions that have an inflammatory component and potentially can open new avenues for therapy and early intervention. The idea here with iron supplement back to what Kristen was saying, we also got interested in trying a supplement that is a flavonoid present extracted from parsley's, and that's shown here in the right, just to have a flavor for what we can do using this system. We used our FACS assay to detect a decrease in this inflamed astrocytes using this compound, and for both ion beta as well as after TNF-alpha treatment. So that's kind of where we're going now. And I'd like to acknowledge Rusty Gage and the Gage Lab here at the Salk Institute, specifically people that worked in this project, Shani Stern, Krishna Vadodaria, Renata Santos, and Ani Sankar. I'd like to thank the NIMH consortium. We had a lot of discussions using those cell lines. And I also like to thank psychiatrists, John Kelso, Martin Alda, patients and family, and big shout out for this very talented group of women that I have the privilege to work with, Ariana, Ruth, Anna, and Vipula. Without them, you would not be seeing such cool data. So thank you so much.
We have time for a few questions. Any burning questions? I guess the logical question would be, is your expectation that anti-inflammatory drugs, if given to bipolar patients, might be therapeutic?
We would like to at least test that in vitro, and also see what would be the effect on neuronal activity too, and how those things would go together. Yep.
Right. It's very exciting. Any other questions that you'd like to ask Carol? Okay, thanks so much, Carol.
Thank you.